Transfection efficiency and optimization. Transfection is the process of introducing nucleic acids or proteins into eukaryotic cells using physical or reagent-based methods. The delivery of protein-encoding DNA expression vectors facilitates protein overexpression, while the introduction of gene-specific microRNAs or other small RNAs silence gene expression and knock down protein expression. Transfection of CRISPR-Cas9 components is also a commonly used molecular technique for powerful and highly precise genome editing. A desirable transfection method reliably delivers the nucleic acid with high efficiency, broad-spectrum application for gene expression, knockdown, or targeted genome editing, all without impacting cell viability. There are two types of transfection. Stable transfection, where the DNA integrates within the cellular chromosome. Transient transfection, where the DNA does not integrate with the host cell's genome. Similarly, viral transduction is another common method that utilizes viruses as the delivery vehicle for the nucleic acid payload. Advantages of stable transfections include persistence of the transfected material and presumably protein expression even after cell division. However, poses significant risk of disruption of essential host genes with genome integration. Transiently transfected material does not integrate with the host genome and is subsequently lost in daughter cells. The decision for choosing stable versus transient transfection is in part based on the duration of the experiment. The hydrophilic nature of the DNA backbone presents a challenge for efficient delivery beyond the hydrophobic center of the cell's membrane. Overcoming this barrier is achieved by two distinct methodologies. 1. Utilizing a physical method, such as electroporation or microinjection, that temporarily perturbs the cellular membrane and allows passage of the nucleic acid. 2. Utilizing a reagent-based approach. Common transfection reagents include calcium phosphate, which involves the uptake of a calcium phosphate DNA coprecipitate. Liposome-mediated, where the nucleic acid is encapsulated in a cationic liposome and is transported entirely inside the cell by a process called endocytosis. Or the use of non-liposomal polymer-based reagents, such as the X-TremeGENE 360 universal transfection reagent, which efficiently delivers a broad range of macromolecules into both easy and hard-to-transfect cell lines. Transfection reagent selection is a critical decision to ensure maximal transfection efficiencies and functionality of your nucleic acid material with minimal cytotoxic effects for the host cell. Because the best reagent often varies depending on the cell line, both the reagent selection and the precise protocol are determined empirically through careful optimization. Prior to optimization, it is important to note that sterile, high-quality DNA or RNA must be prepared. A common and effective method for optimization is the use of multivariate approach in a 96-well plate. Optimization should include, but is not limited to, investigating the following parameters: DNA or RNA concentration to transfection reagent ratios, the amount of time for both DNA reagent complex formation and incubation with your cells, and the transfection reagent itself. Successful transfection optimization would include identifying critical parameters that result in maximum transfection efficiency, significant gene expression or knockdown, while maintaining high cell viability. Due to the minimal cytotoxicity of the polymer-based reagents, like the X-TremeGENE 360 transfection reagent, both the reagent selection and optimization procedures can be dramatically reduced. With the X-TremeGENE 360 transfection reagent, you do not need to optimize the medium exchange step after addition of transfection complex. This universal transfection reagent removes much of the guesswork as it is reliable for transfection of DNA, siRNA, miRNA, and CRISPR-Cas9 components for both easy and hard-to-transfect cell lines. Because transfection reagent selection is often dependent on cell type, we recommend using our transfection reagent selection guide and our technical support team to select what products are right for you at sigmaaldrich.com/extremegene.